Hi, I'm Erin Gray, an assistant professor of aquatic genetics at the University of Maine. I'm really sorry I couldn't be there with you today to talk about eDNA, but I'm happy to make this recording talking about eDNA sample processing and data considerations, or what to do with your water sample once you collect it. Here's an overview of what I'm going to be talking about today, and this is not an exhaustive list of all aspects of sample processing and data considerations, just the main points that you have to think about when you're starting to plan your study. Okay, so after the first two sessions of this workshop, I'm assuming that you all designed a really optimal survey and you went out and you collected your water samples. And I'm going to talk to you about what you do with those water samples now that you got them. Now, the vast majority of times, you're going to want to filter those water samples through a filter membrane. And before you do that, you want to choose a filter type. Your options include the material of that filter. So common materials for eDNA are glass fiber, cellulose nitrate, or CN, or polyether sulfone, or PES. PES filters are sometimes called SUPOR filters. You're also going to want to choose a pore size. So pores are just the size of the hole in the filter. You don't want your pores or holes to be too big, or else the DNA is going to just go right through there and not get caught in the membrane. But you also don't want the pores to be too small, or else your filter is going to clog and you're not going to be able to process the volume of water that you want. So how do you decide what material or pore size your filter should be? I suggest that you just check the literature for your particular study organism or organisms and your habitat. So go to Google Scholar, search up eDNA, your organisms, your habitat, and see what people have used in the past. There's a good chance that that's going to work for you too. And then you probably also want to try out 1 or 2 or 3 different filter types just to make sure that they'll work for you. Once you choose your filter type, you then have to think about how you're going to get water through that filter. And there's 2 main approaches for this. The first is that you can push water through the filter, and I'm showing you an example of that here. In this example, we have a filter cartridge. This is a Sterivex type of filter cartridge, where the filter is going to be on the inside surrounded by a tube. This tube connects to a syringe, so you can get your sample into the syringe, connect it to the filter cartridge, and push it through, where DNA is going to collect on that filter. That definitely works. The other approach you can take is that you can pull water through the filter. So here I'm showing you 3 different ways to do that. The first way is a pretty standard lab vacuum pump setup, where we have our vacuum pump, which just connects to an outlet. This could also be a peristaltic pump. This is going to create a vacuum. And over here in this first filter flask, we have a filter membrane on the bottom. Water goes in over top of it, gets pulled through, DNA gets caught in the membrane. Just remember, if you have one of these setups, always set up a moisture trap or an intermediary flask to make sure that water doesn't get into your pump. And then they now sell these fancy field vacuum backpacks that are battery powered, so you can actually suck water up and filter at the same time in the field, which is pretty cool. You could also just buy a hand vacuum pump at your local auto parts store if you want to go that route. So how do you make your decision about which filter type, whether to push or pull or what have you? It just kind of depends on your values, because all of them have trade-offs. So there's trade-offs between volume, time it's going to take you to filter that volume, cost, field versus lab. So for example, the backpacks are really nice, but they're pretty expensive, so you might have to go with something more affordable for your case. It's all just something that you have to make a decision about. When you're filtering, you're also going to want to include negative and positive controls at this stage. The negative controls are going to detect contamination that could be happening between your samples being filtered, and the positive controls are going to detect failed filtering, for example. For negative controls, you're going to want to filter pure water, or as pure water as you can get, either in the lab or the field. And for positive controls, you can filter real or mock positive samples. As soon as you filter your samples, you're going to want to store those filters in a way that reduces DNA degradation. And again, we have several options for this. The first one is freezing. If you have a -80 degrees freezer, you can just put your filters in there. The only thing to think about is that you want to minimize freeze-thaw cycles that will degrade DNA. Another viable option is dry storage. So in the absence of water, DNA degrades very slowly. So you could, for example, put your filters in a bag with silica gel beads and they should be fine for a very long time as well. In the field, a lot of people opt to store their filters in some sort of preservative liquid, either buffers or ethanol. And before I go into these liquids, I just want to say that if you're storing your filters in the liquid, you should try to use a sterile DNA low bind tube that's going to reduce the amount of DNA that sticks to the side of the tube, which is a good thing. So if you choose to go with a buffer, the logic behind buffers is that you're going to include some chemical that's going to reduce the effectiveness of DNases. So DNases are enzymes that degrade DNA. One of the most common chemicals that does this is EDTA. If you'd like to make your own buffer, there's several recipes out there. Just Google, for example, TE buffer, low EDTA, or Longmire's buffer. Those have all worked well for me. Or you could buy your buffers online, and here's just a couple options. Ethanol is also a pretty good DNA preservative, although I will say that ethanol is flammable. So if you want to mail your samples, you're going to run into problems with that. So if you want to mail your samples anywhere, I'd recommend using a buffer, dry storage, or freezing. But ethanol is a perfectly fine way to store your filters. If you use it, I recommend using 95 to 100% non-denatured ethanol. Denatured ethanol just simply has additives to it, and usually these additives are okay, but sometimes they can mess with things downstream. Now that you have your filters safely stored away, I'm going to assume for this workshop that you're going to send those filters to a lab to have them do the extraction and run the assays on those samples. So here's just a quick checklist of things to think about when working with labs. The first thing to ask yourself is, does this lab work with environmental DNA? So to give you some background, a lot of labs are going to do sequencing and PCR, but I'd say the majority of them are working with pretty clean samples that come from tissues and things like that. Environmental samples have like low amounts of DNA, a lot of inhibitory substances. They're just pretty different. So you're going to want a lab that has experience with environmental DNA samples. The second thing to ask is, do they offer the assays that you want? If you want digital droplet PCR, do they offer that? Do they offer metabarcoding? Things like that. The next very important thing to ask yourself is, what are their quality controls? Do they, for example, do their extraction and PCR in separate rooms? They really should, because this reduces contamination quite a bit. Are their rooms and equipment sterilized on a regular basis, and how so? Very important is, how are samples and data managed and stored? I had, for example, a lab lose my samples, which was not great. Another thing to think about are, what are their processing controls? Do they include, for example, PCR blanks or no template controls as a routine? They probably should. And also, how many PCR replicates do they typically do per sample? Other practical considerations are things just like timeline, like how fast can I get your samples done? Days, weeks, months? How will the samples be delivered or returned? If you have to ship them really far, frozen, don't ship them on Friday, right? Because they'll sit over the weekend and things like that. Another thing is, how is data going to be transferred to you? This can really matter if you do metabarcoding, because you can't just email that data. You have to figure out a way to transfer it to you. Also, every lab should give you a quote for the work that they're going to do, and they should also be able to answer all your questions pretty well. And it bears repeating that at this step, you also want to think about controls. Again, make sure the lab is going to have some sort of negative control to detect contamination. For example, if they're doing PCRs, they should do a PCR with just blank water in it to check for contamination. They should also do positive controls like synthetic gene, sequencing standards, and things like that to make sure that the assays are working as intended. Without a doubt, the most common question I get about eDNA is, how much does it cost? And that's a really hard question to answer. And it's hard because it depends. It depends on your samples, how many you have, how you want to have them assayed, and it also depends on the lab. So labs have different price structures. They run different assays. So it's really hard for me to give a sort of general cost per sample. But what I'm going to try to do here is walk you through a couple scenarios of a couple different assay types and what they would cost at the Maine eDNA core facility. And in the first scenario, I'm going to assume that you have filtered 20 samples, 16 samples from a stream, for example, 2 are field controls, 2 are filter controls. And what you want to do is you want to detect and quantify Atlantic salmon DNA from these samples. Now, if you go to the Maine eDNA Core website, you'll see that they offer both qPCR and ddPCR assays for this task. Here is a portion of the service rate sheet for the Maine eDNA Core for clients that are external to the University of Maine. So if you are from the University of Maine or you're working with somebody from the University of Maine, your prices will be a little bit cheaper, but about the same. You can download the full service rate sheet at the link below. And I'd also like to note here that these estimates I'm giving you are just estimates. If you want a quote for your project, you should email the director, Geneva York, at the address there. Now, I'm going to assume that you shipped the eDNA Core your filters that were either in buffer or frozen. And if that's the case, then the first thing that the lab's going to do is they're going to extract the DNA from those filters. And right here, I'm assuming you sent them 20 filters, and then the lab's also going to do a blank extraction negative control for a total of 21 samples. Now, to find the rate for this, you go down to eDNA extraction, and the typical eDNA extraction protocol that the lab runs costs $8.50 per sample. If you want your extraction done another particular way, you just have to work out that rate with the core directly. So if you multiply 21 by 8.5, you get about $178 for DNA extraction. Now, the core recommends that you do a cleanup of your extraction, and this will just remove inhibitors that might get in the way of the PCR reaction. This is an optional step, but it's recommended, especially for environmental samples where inhibitors are common. So this extraction cleanup step costs $6.25 per sample, so about $130. Now, I mentioned before that Maine eDNA has two different assays that you can quantify Atlantic salmon DNA with. The first assay is regular qPCR, and to find the rate for qPCR per sample, you would go down here. And in this line, it says targeted eDNA single probe qPCR assay. That word probe tells me that they're using a TACMAN probe, which is the state-of-the-art technology for qPCR, so that's a good thing. And then I come over here and the unit type is per sample, and I see that the rate per unit is $26, which if you've done qPCR before, that seems like a lot. However, the thing to note here in additional info is that this rate includes 4 replicates of each sample. So they're going to include the replicates for you, so that's nice. They're also going to include the standard curve and the negative control. So 26 times 21 comes out to 546. Now the other assay you could choose would be digital droplet PCR. And Maine eDNA has an assay for Atlantic salmon digital droplet PCR, and their general ddPCR rate is up to 8 samples. So they run them at 8 at a time. So it's going to cost you $125 whether you run 1 sample or whether you run 8. Now you see that that per sample rate is a lot higher in ddPCR, but the key thing about ddPCR is that you don't need to replicate. And I'm going to talk about that more in a second here, but just know that it's just 21 samples, and you can fit up to 8. So assuming that we're going to have some blanks and some standards, but we don't need replicates, that brings us up to about 24 samples, maybe a little bit more. So maybe we'll do 4 batches of 8, and that'll get us up to $500. So you can see that qPCR and ddPCR come out to about the same. Okay, let's think about another scenario. In this scenario, we have the same number of samples that we filtered, 20, except this time, instead of detecting just Atlantic salmon, we want to detect as many fish as possible. Maine eDNA has a 12S meta barcoding for this task. So this is how you would break down the pricing for meta barcoding of your 20 samples. So the thing you'll note here is DNA extraction and cleanup is the same. Basically, you just have to get the DNA nice and clean out of your filters before you can do anything, whether it's a PCR assay or meta barcoding. But then for meta barcoding, what you're going to want to do is prepare your samples to run on the sequencing machine. Now the Maine eDNA Core Lab normally does a 2-step library prep. Sometimes you might hear people talk about a 1-step library prep. Maine eDNA Core usually does a 2-step, but you can ask about a 1-step if you'd like to do that. If you go down here, their library prep 2-step process costs $500 a plate. A plate, by the way, is 96 samples. Now the Maine eDNA core will split plates, so you can pay, for example, half a plate if you only have 48 samples, or in our case, we only have 21 samples plus a couple of controls, so a quarter of a plate. Not all labs are going to split plates and things like that, so it's important to see if they do. But anyway, so assuming that we can split our library prep by a quarter, it's going to cost us $125. And now the MySeq run, and by the way, this is just the time for the technician to run the MySeq is $300 per plate or per run. So assuming that you're going to split this run again into a quarter, it's going to cost you $75. Now it's important to note here, and this is something that's in additional info, and it unfortunately got cut off here, but the sequencing kit and reagents are not included in the MySeq run in the core's service rate sheet. So right now, these reagents are going to cost about $600 for a quarter plate. And they'll usually be about this, but it does vary a little bit because it's basically what the company charges. So all in all, if you got a quarter plate of samples to MetaBarcode, it's going to cost you about $1,100. Now, if you're splitting your plate, you might have to wait a little bit longer because the core is going to wait until it has the other 3/4 of a plate to run your samples. Hopefully that made sense. Now for this last section of the talk, I just want to walk you through really quickly the different types of data that you can expect to see from these different assays. We'll start with qPCR first. So if you remember Karen talking about the PCR process, remember that we start with a single double strand of the DNA molecule, and then that strand gets copied. Each cycle of PCR, it gets copied. So the first cycle we start with one double-sided strand. After the first cycle, we have 2, then we have 4, then we have 8, and then it gets exponentially more and more and more with each cycle. So what if we could make that DNA glow? And if we could measure that glow at each PCR cycle, we would expect to see the glow increase with the copies of DNA, right? So here I'm showing you just that. One type of qPCR, we have a molecule that only glows when it binds to double-stranded DNA, as you can see here. So before the first cycle, we just have 1 copy, so we have 1 unit of glow. At the end of cycle 1, we have 2 units of glow, then we have 4 units of glow, then 8, then 16, and so on and so on and so forth. And if we were to plot this out with cycle number on the x-axis and glow level on the y, we would see an exponential increase in glowing. In a qPCR assay, what we're going to do is we are going to measure glow, shown here as delta Rn, over many cycles. And in an eDNA assay, 50 cycles is actually pretty common. But here I'm just showing you 40 cycles. And what you're going to see is if we have your target DNA and we're copying it in the PCR reaction, you're going to see glow be very low for a few cycles. And then all of a sudden it's going to pop up, right? And this is the exponential growth where millions and millions of copies are being made. The glowing's getting really big, really fast, and then it's going to level off. And this leveling off just means that we've run out of reagents to make copies. So it's just going to plateau. And what I'm showing you here on this top one is qPCR curves for positive controls. So these positive controls have known amounts of your target DNA. And you can make these positive controls either by diluting tissue extractions like that you got from your target organism, and you know, for example, this is Atlantic salmon DNA, or you can order them online now. People will make your target gene for you and ship it to you in the mail. It's pretty crazy. But anyways, you'll make different dilutions of your target DNA. So here we have many copies, 1 × 10⁶ copies of your target gene. This is a little less, ×10⁵, and down here we just have 10 copies of your target sequence. And what you'll see here is that when you start with more DNA, like in this red curve, you pop off faster. You get exponential growth faster. And then you pop off slower the less and less DNA that you have. So we use this sort of the cycle at which your fluorescent glow starts to really increase exponentially, we use this as sort of a measure of how much DNA you started with in your sample. And this measure we call the CQ or the CT value. And it's basically just when your glow level passes a certain threshold, what cycle was that? So you can see here in this red curve, it started to pop off around 17, so they marked it as 17.5. And the yellow was more like 20, and the light green, 22.5, and so on and so on and so forth. Okay, so when we make this plot, we typically plot the dilution or the number of copies on the log scale, just because you usually start with like 1,000,000, right? And then you go down to a 1 or 10. So you have to log that in order for it to make sense. And when you log it, you usually come out where you can fit a nice straight line relating the copy number or the CT value to the quantity of DNA that you started with. Now, making one of these standard curves is really useful because remember, your field samples, you don't really know how much DNA is in there to start with. But what we can observe if you run that PCR reaction is we can observe the CQ or the CT value, or when, like I'm showing you here, this is our field sample in the dotted black line, when this curve starts to pop up. And here it's about 31, okay? So what I'm going to do is I'm going to observe the CT value of my unknown field sample, and then I'm going to go plot this value onto my standard curve. So observing the CT value, I'm going to plot it. It was about 31, okay? So where does that put me in terms of quantity of DNA? Well, it puts me between 30 and 100. I'd say about 50 on that, okay? And so note here I'm saying quantity is pretty vague. You can measure quantity as DNA copy number if you order it from a company, or if you just have sort of known concentrations of DNA that you extracted yourself, you can use quantity as nanograms per microliter. And just FYI, some labs will do these calculations for you, so they'll do the standard curve fitting and all that and estimate it yourself. Other times they're just going to give you the CQ values or the CT values, and you're going to have to create the curve and infer the samples yourself. Now, ddPCR is conceptually similar to qPCR in the sense that copied DNA is going to glow, except in this case, rather than mixing all the DNA together in one tube, we're going to separate each DNA molecule into separate droplets. And in each droplet, we're going to have one molecule. And if that molecule happens to be our target DNA, it's going to copy itself and glow. If it's not our target DNA, or there's no DNA molecule in there, there's going to be no glow. And the ddPCR machine is going to measure the number of droplets that are glowing versus the number of droplets that are not glowing. So here you can just see these would be the droplets that are glowing that it measures, and these are the ones that didn't glow. So you're just going to get a proportion of droplets that had the PCR reaction occur versus the proportion that don't. So this data is a lot easier to interpret. It's just the quantity of DNA in your sample. You don't have to do a standard curve or anything like that. However, it is a good idea to put a few standards, a few blanks, a few samples of known amounts in there just to make sure that the reaction is going as designed. The last thing I'm going to talk about in this presentation is the type of data you can expect to get back from a metabarcoding assay. And this is a big, busy slide, but if you only take away one thing, I just want you to know that you're going to get a lot of data. Specifically, you're going to get 1 or 2 very large FASTQ files per sample. Now, these FASTQ files are going to contain millions of raw sequences we call reads. I usually get about 20 to 25 million reads per FASTQ file, for example. Now, each of these reads is going to have the name of that particular read, then the sequence of bases that the machine detected, so about 100 to 300 bases of A, T, Gs, or Cs. In addition to the bases, you're going to get the base qualities. So basically, how sure the machine was that each base is what it is. So how sure was it that this G was actually a G? How sure was it that this A was actually an A? And usually it's not very sure at the beginning of the read, right? And so then we'll want to trim off sort of the low quality scores at the beginning and the ends of the read. And then there's a bunch of other steps we do to kind of clean up potential sources of error and try to identify really what true sequences they are. So all these bioinformatics pipelines are trying to do is find the true sequences from the noise. And then once they do that, they're going to try to identify these true sequences by assigning them or matching them to sequences in a reference library. And then hopefully you can get all the species of fish in your sample, for example. Just so you know, you're likely going to need a computer cluster to run these pipelines, just because it's so much data, your normal sort of personal computer might not be able to do it, especially if you have, usually if you have a handful of FASTQ files, you can do it on your computer, but any more than that, you're probably going to want to run it on a supercomputer. And luckily, many labs, like the Maine eDNA Core, but also others, offer bioinformatics services that you can use. So that's the end of my talk. That was a very brief overview of some of the key points and decisions you need to make when processing your samples and analyzing your data. I hope that I'm back and able to at least Zoom with you to answer any questions, but if not, I'm sure that Mike Kinnison and Karen James can answer any questions that you might have. And feel free to email me at erin.gray@maine.edu if you want to discuss anything further.